Thank you, Barbara. Thank you. Thank you all for coming. It's a true pleasure and an honor to be invited in these QAAFI Science seminars. The topic that I'll be going through today obviously relates to taste. That's the main interest in my research group. But taste probably not in the classical concept that most of you would be thinking of. Of course, taste, it's gone somehow a different path to that will probably uncover. It's actually telling us a little bit about how we may be able to control food intake, and that's what I'm going to try to relate it to today: taste and the control of appetite. Nowadays, we are only always looking at rankings, right? All of us want to be top, up there, number one in the rankings. So it seems that, it seems that Queensland is not doing quite bad in this ranking, except that that's probably not the ranking you want to be on top, which is the ranking of obesity. It is true that we're doing quite good in Queensland, but definitely the whole world we're doing quite good at obesity rates, and therefore that's the framework that I would like to start my talk today. I would like, and that's my long-term goal, I would like to contribute in the understanding on why we keep getting fatter and fatter as a whole human beings. Now, there's no question that one of the drivers of food intake is taste. So we eat more when we like better our foods. And in fact, there's probably not a lot of shame when we recognize that we prefer to have a good, tasty meal than a maybe not that good, but healthy meal. What do you prefer? Just ask that question. And actually, when we are asked, we say, yeah, probably I prefer a tasty meal, right? But the question today, and the question I want to ask you, is what's tasty to you, right? Well, tasty, yeah. So if I start asking each one of you, what's your favorite food? You're probably all going to tell me something different, right? And yeah, we have different cultural backgrounds, that's true, but even siblings, people in the same family, we have different food choices. And why is that, right? So that's one of the questions that I'm going to try to answer today. And the point that I'm trying to make is that when it comes to flavor perception, we're not equal. In fact, we're quite different one from the other. We have been learning about some of us being hypersensitive to some flavors and to some tastes, while others are just normal or hyposensitive to those. The extreme cases would be, as pointed here, anosmia, which an meaning lack of smell. So there's some people, practically most of us, we will be probably missing the capacity to smell one compound or another. That's an anosmia for that specific smell. When that refers to taste, then we talk about ageusia. So people that cannot detect some taste-active compounds, they suffer from ageusia for that specific taste. So those being the extreme, anosmia, ageusia, there's everything in between, from the hypersensitivity to the no sensitivity at all. Now, if we look at something relatively easy that you may think, well, we're not that different. When it comes to detect different tastes, I'm talking here about 4 of the 5 primary tastes: sweet, salty, sour, and bitter. And those are just a small number of panelists, and those are the ratings. And you will see that the difference in concentration that different panelists will detect may be just a par by 100-fold. That's pretty good. Meaning that some of us will need 100 times more sugar than others in this room to detect the same amount of sweetness. 100 times is pretty good, right? It's not a small one. If you look at salt, similar rates, sour, same, bitter, the same. Actually, that's umami. For those of you not familiar with umami taste, umami is in humans at least, is related to monosodium glutamate, glutamic acid. That's umami, or savory if you want. Same case here. So big differences in the way we sense umami, in the way we sense sodium. Now think about, think about sodium and think about hypertension. If some of us keep adding salt into our plates, why is that? Well, maybe because we can't sense it as well as others do, right? And that's where we're trying to understand, and what's the impact of this variation, diversity in taste and smell sensing. Now, if we look at the physiological drivers of this diversity, there's no question there's a lot of potential different causes, from whether we just had the meal and we're satiated, oral hygiene, disease stage, age. Unfortunately, we get older, we're not getting better at taste perception. Dehydration, important. But today I would like to focus in what's in our genes. What if a good part of this diversity of taste perception is due to variations in our genes, what we call genetic polymorphisms? Now, let me just briefly introduce you a concept that for probably a number of you're already familiar with, which is the SNP, the single nucleotide polymorphism. Well, you know that our genetic material is, if you want, packed in chromosomes. That's the chromosome here. When a gene is to be translated into a protein, it will first unfold, and then the gene will be transcribed into what we call the messenger RNA. Now, what's a gene? A gene is nothing else than a sequence of bases or nucleotides. And that sequence of nucleotides is very specific for each gene. So we have to keep that TGCAGT to define that specific gene. If that sequence changes the gene, right? It's very simple. So actually one of the ways nature adapts to new environments is by mutations. And sometimes these sequences changed. And the change of one nucleotide, if it's passed to the next generation, will become, therefore, a polymorphism. So we have one gene with two different sequences. Even though the sequence is only changed by one nucleotide, this change in one nucleotide may have an impact on the type of protein and the functionality of that protein, right? So that will be the case. Therefore, when one of the amino acids incorporated into the protein changes from one type to another type through the ribosome tRNA system. Now, we have lots of examples of these type of SNPs, single nucleotide polymorphisms, affecting food perception. One of that's been mostly studied the most is androstenone, which is a pheromone that's produced by the pig. And this pheromone actually is lipophilic. So every time we eat a pork chop from a male that has not been castrated, we probably have some androstenone in the fat. Now, a few of us, or some of us, part of our population, is very sensitive to androstenone, and part of the population is just not sensitive to androstenone. So the bored taint is just happening to part of that population. And we've gone down to the gene, this olfactory receptor gene, and we now know what polymorphisms are related to the sensitivity to bored taint. There's a number of other examples here, cilantro versions, or the methanethiol, which is the asparagus smell when we eat asparagus, and the metabolite of, it has very characteristic smells, but there's part of the population that will not detect that smell. So if we start looking at all these genes that may have polymorphisms in our population, think about how many different combinations we may have. This is a list of chemosensory genes that we're becoming familiar with. Bear with me, humans, we have 388 active olfactory receptors. And it's becoming quite clear, although we do not know the repertoire yet, but it's becoming quite clear that each one of these genes will have polymorphisms. So the combination of these 388 genes makes us, literally each one of us different. We do have different capacities to smell and taste. I am actually going to shift my talk, and I will be focusing in taste receptors. That's really the area where we are working actively. And for two reasons, basically. One reason is the number of genes involved is much smaller. So we're not talking about almost 400. We're talking about, 28 here plus maybe another 20, around 50 genes. The other reason that I'm particularly interested in taste receptors is because taste relates to nutrients, and I'm going to further explain that in another slide. But just a very brief reminder, I'm sure you all know the basics of taste perception. So we have the taste papillas in the tongue, and within each one of these taste papillas you will find somewhere between a few to several hundreds taste buds. The taste buds are onion-shaped type of organelles that contain the taste sensory cells. There's a number of different types of sensory cells, which I'm not going to go in detail. What's most relevant is understanding that the taste sensory cell is a quite unique cell, and I'm going to be talking about this type of cells later on my talk. So that's why I want you to please pay a little bit of attention, because it has some microvilli, so it gets in contact with the surface of the tongue. That will be the surface of the tongue. That's the microvilli where the taste receptors will be. And then it has the capacity to form synapses with neurons. That's very unique. It's a dual cell from that perspective. And we'll further talk about this type of cells. Now, what we've learned up to date on taste, on different taste sensing, and I'm coming back to my comment on my interest in nutrients and nutrition, being nutrition is my background. The truth is, taste has evolved to monitor or to analyze the nutritional content of the diet. If you think about what's sweet, really what's sweet is simple sugars, is the carbohydrate part of the diet, right? If you think of umami, I said, in humans it's quite narrowly tuned to glutamic acid. In other animal species, actually it's widely tuned. In laboratory rodents, the umami taste receptor will actually be stimulated by 18 or 19 of the L-amino acids. So almost any amino acid will stimulate the umami receptor. When we look at salt, it's sodium electrolytes. Sour for organic acids. Fat is an interesting one. We cannot really call fat a taste, because one of the principles of defining a taste is you need to be able to describe a taste different from other taste qualities. And we have a problem in defining how fat really tastes. But what it is true is that we are capable of sensing fatty acids in the oral cavity. So we can call fatty acid a chemosensing capacity of the oral cavity if we don't want to call it taste, right? There's other potential taste types that are being discussed if you want. Calcium could be another sensing in the oral cavity, if not a taste. Bitter is it's a big family of taste receptors that's actually focused in finding potential toxic compounds in food. So that's why it just gives a rejection response to a given food. At least when we're infants, then we may learn to appreciate bitterness. I'm thinking of coffee or other drinks, but that's because of we're learning about a bitter taste non being toxic and therefore being acceptable. But initially, the innate response to bitterness will always be rejection, because it's potentially a toxic compound that could kill us, right? And so on. There's been talks about a taste on carbonation, which in fact is a combination of somatosensing and sour. etc. So there is a number of different tastes that you might not be familiar with, but they are all related to nutrients that we are ingesting through our diets, right? Now, it is important to add, when we talk about different tastes, I said we need to be able to differentiate the taste from other taste types. That's one of the requirements. But also we need to know the molecular mechanism. Nowadays, if you don't have a receptor, you're not going to be entitled to call a novel taste. So if you want to discover a novel taste, you bring a receptor and bring a different sensory quality, and then you're all right. If not, you're not. Now, so about receptors, we've learned about the T1R family, which are 3 genes for sweet and umami. We've known about the T2R family, 25 genes for bitter taste. A number of receptors involved in fatty acid sensing, a number of receptors involved in amino acid sensing, umami, savory, and then salt, which is a channel, and sour. These are the type of drawings that will summarize what's a channel for sour and salty. These are the T1R for sweet and umami, and this would be for bitter taste receptors. Now, the sweet and umami receptors have something unique, which is a huge extracellular domain, which just sticks out of, if you want, the surface of the tongue. That's where the food debris will come in contact. How do they come in contact? It's been defined as a Venus flytrap type of model. So we have a huge domain, the extracellular domain of the receptor, which is a 7-transmembrane receptor, GPCR. And this extracellular domain actually is activated by your sugar, your active substance. When the active substance, say the fly, gets into the active domain, then the Venus flytrap just closes, and so does the taste receptor. It closes down, changes conformation, and that will give the depolarization of the taste cell, and from here to the brain, and then taste is being sensed, right? Now, the understanding of this mechanism is it's actually been having a big impact on the food industry. The food industry has been looking for non-caloric sweeteners probably since 1880, when saccharin was discovered as a serendipity. And ever since, the food industry has been looking for alternative to sugar, non-caloric alternatives. And as a result, today there's good dozens of compounds which have various type of potency as sweeteners. But the food industry keeps looking for the perfect sweetener. Why? Well, because none of the sweeteners that are today in the market have the pure sugar taste. And that's compelling. And the reason is molecular. I just took this from a question I had prepared for one of my lectures. And it's just to bring you to the attention that the food industry has been heavily involved in this type of research, understanding the molecular basis of taste so that they can develop novel sweeteners with the pure sugar taste. Now, I could use any of the colors in the market. They all have all these different names with different profiles, which are all very similar, classic, next type, 1, 0, whatever. And always is looking for a decrease in calories from a lower to a 0 calorie, decreasing sugars. And what's been recently being quite a sort of a solution for these industries, it's been not reducing sugar to 0, but keeping some sugar. And why is that? Well, the reason is because they came up with the model which works both for the umami and for the sweet, where the main molecule, which would be the sugar, will sit at the bottom of the Venus flytrap domain, while there are enhancers of the sweet or the umami that will sit just a little bit outside that cave. Those are the enhancers, and that's what the industry has been working on. Rather than working now in trying to substitute the sugar for something different, they are now trying to look for a sugar enhancer that will allow the substantially decrease the amount of sugar in a drink or in a food without losing the actual pure taste of sugar. Now, the reason is because all the other main non-caloric sweeteners, not all of them, but most of them, they do not interact directly with the active binding site. They interact allosterically with the receptor, and therefore they interact with other receptors as well. Saccharin, for example, interacts with the beta receptor. So that's why we have this aftertaste with saccharin. It's probably related to the bitterness of saccharin itself. Now, that's what the solution, and you probably will see that in the market in the near future, sweeteners that are actually enhancing sucrose rather than replacing sucrose, right? And that's all based on these computational models. Now, something that may have escaped to your attention because I was just passing through very quick, but I was emphasizing the fact that when it comes to cola, I would bet that each one of us have a different perception of cola. Why? Because it involves a number of different tastes. We've said sugar, yes, but it's got quite a significant amount of sodium, so it's a bit salty. It has caffeine. Caffeine is bitter, so actually it does play with bitterness as well, but also has a non-caloric sweetener. It has carbonation. Carbonation also stimulates acidity, sour, and you have citric acids and phosphoric acids. So it's really a cocktail of tastes that we have in cola, and that's probably why some people would say, oh, cola, I can't drink it, it's just too sweet, or no, it's just too acid, because we do have different perceptions of the same stimuli. Now, I'm going back to the polymorphisms because that explains individual differences, and that's probably one of the main messages of my talk today. The fact that what probably shapes the way we taste foods is the divergence coming from polymorphic genes. We know that we have the umami taste receptor, the T1R family in general. Any of the 3 genes we have already found in the population changes or polymorphic different alleles being present. But also bitterness, bitter compounds, each one of them, we're finding that there are a number of polymorphisms, alleles that makes us taste bitterness differently. And in fact, let me just stop here for a while in the sense that I'll just further explain this number 38. Keep that number because it's going to be coming back and forth several times in the next few slides. Number 38, this bitter taste receptor that we call number 38, is probably the one that's been studied the most, and I will explain the reason. Well, the bitter receptor 38, we know that there are 2 main alleles in our population, which accounts for 90% of the population. And the difference between that gene in these 2 alleles is 3 SNPs, 3 single nucleotide polymorphisms, a proline that went into an alanine into a valine, and a valine into an isoleucine. That accounts for the allele PAV or AVI. Now, the PAV, if we are homozygous for PAV, so mom and dad gave us that PAV gene, we are likely to be hypersensitive to that compound here, propylthyouracil, which is a bitter compound. It's a synthetic compound, very similar to the thioureas present in broccoli and in other vegetables. In fact, it has been used to study why some kids do not eat vegetables, trying to relate it to the sensitivity to thioureas, right? Now, if we happen to be PAV, which I'm not, you will be a hypo-taster. So if I give you a very small amount of propylthyouracil, you'll remember my name, right? Because you're really going to suffer bitterness that it's very unpleasant. If you're a heterozygous PAV-AVI, which is one of each alleles, then you're a normal taster. And if you're a homozygous for the AVI-AVI, you just simply do not detect unless there's very high amounts of prop. So you're a non-taster or a hypo-taster, right? Now, and that's all fine, that's fun, except that what we're just finding, and I will further explain not only with this gene, but with other genes, it is becoming more and more clear, I would say, with robust evidences that, the sensitivity to some of these tastes, is related to the ability to control food intake. So that's two different works pointing in the same direction, saying that if you're a hypo-taster or a normal taster for prop, in fact, you're doing not that good in controlling your consumption, and compared to hypo-tasters, you're just over-consuming calories. It seems that your brain is not telling you to stop early enough, and you keep eating. And that's been linked to the sensitivity to prop. So it seems relevant, so it might be worth to keep an eye on that. Now, in our group, we decided that we had a problem accepting that if you're a hypo-taster for prop, that you're just a hypo-taster for everything. Yeah, it doesn't sound like good. So we decided to test the hypothesis that if you are a hypo-taster for prop, you just happen to be hypo-taster for prop. That's it. Another compound with a different receptor will just be a different story. That was our hypothesis. So what we did is we chose 8 different beta compounds. The reason why we chose them, well, first of all, hopefully they were non-toxic and natural compounds, and they had been approved by the FDA and so on. But the main reason we did is because we wanted to check different beta compounds that were sharing taste receptors and different compounds that were not sharing taste receptors. See if there was any correlationship in the sensitivity to the beta taste of those compounds. So as you see here, if you compare binary combinations of these 8 compounds, some of these combinations, actually half of them, had no taste receptors in common. They were just going through different paths of bitterness perception. Some had 1 taste receptor in common, 2, 3, 4, and even the caffeine versus quinine had 5 taste receptors in common, so very similar. Our hypothesis was, look, if they do not share taste receptors, the sensitivity to those compounds should be independent. There's no reason why it should be related. And the opposite is true. If they share taste receptor, their bitterness might be related within any single panelist. So we went through the labeled magnitude scale from barely detectable to strongest imaginable. So usually you would expect that a hypersensitive person will be ranking the bitterness in here, in the very strong to the strongest imaginable. And then the normal would be right here, and the hypo-tasters would be right there in the weak or no. Let me jump in the last third of my presentation, switching from bitterness and bitter sensitivity to fat sensitivity. Big topic, very important for obesity, without doubt. This is just to sort of illustrate that over the last 6-7 years, we've learned a lot about how fat is perceived, and particularly fatty acids. Fatty acids are perceived through a number of genes, and therefore that opens also the door to understand fat sensitivities and how fat might be perceived in different individuals. Now, again, when it comes to some of these fatty acids, it's been well described that part of the population is hypersensitive to oleic acid, while part of the population is just simply has a very low sensitivity for oleic acid. And that's fine, except that, another interesting correlation. If you happen to be hyposensitive to oleic acid, you will have a tendency to have a much higher intake of calories than if you are a hypersensitive person. Now, Where do these calories come from? Mainly the differences were in meat and in dairy. Those are the main differences. So it seems somehow that if we're capable of sensing fats, that will give us better clues from a metabolic standpoint to respond in that hunger satiety cycle and decrease somehow food intake compared to a hyposensitive person. And why is that possible? Well, probably the answer is outside the oral cavity, includes the oral cavity. But what's unique about what we're learning about taste receptors and taste sensory cells is that they are not only present in the tongue, that's a taste bud in the tongue. We're using here a biomarker, which is this protein here. We call that gastacine, and gastacine is an intracellular protein which is very specific for taste sensory cells. You have gastacine, you're a taste sensory cell. So if we mark gastacine, you will see taste sensory cells in a number of epithelia in the GI tract. Those are photographs from small intestine in the rat. Now, what are these taste cells doing here? Not only in the small intestine, but now we know that all these different taste sensory cells, all these different receptors that I've been talking about, they're all present in a number of different tissues. Remarkably, the stomach being particularly rich in T1Rs and T2Rs, but also fat sensing cells. That's the small intestine, in the pancreas, in the liver, large intestine, and ceca, different parts of the small intestine. Now, the hypothesis behind what are taste sensory cells doing in the GI tract is that, well, they're actually doing, possibly they're doing the same they're doing in the oral cavity, in the sense that they are monitoring nutrients. So they are advising everybody else, there's nutrients in the gastrointestinal tract, there's nutrients coming, get ready for digestion, absorption, and eventually kicking through the anorexic path of the hunger satiety cycle. We had enough nutrients, stop eating. That's going to be the signal. How is that happening? Well, because they are expressed by cells that we identify as endocrine cells in the GI tract, and therefore the stimulation of the taste receptor will lead to the increase in the release of incretins such as GLP-1, but also anorexigenic hormones like cholecystokinin, among other hormones. Now, so our understanding today, and trying to wrap up a little bit on that part of the presentation, is what is that taste sensitivity have to do with appetite? Well, we believe that if you have a high sensitivity for a given nutrient, and it changes from nutrient to nutrient, and that's the complexity, and that's where we need to advance. But if you have a high sensitivity for a given nutrient, that high sensitivity will give strong signals back to the brain, so that the brain makes decisions, right? That's what the brain's supposed to do. Whether it's coming from taste or it's coming from the GI tract, the decision that the brain takes, if we have a strong signal, will be clear and cut and quick. However, if we have a low sensitivity for that specific nutrient, what happens is that the signal back to the brain, it's either not there or it's very weak. So that the decision making on all the hormonal cascade that will lead to satiety is much lower. We need more quantity, volume of the same nutrient to get the same response from the GI tract. And that's been related, therefore, with lean and obese individuals. Basically, in the cascade of having high sensitivity, the likelihood of reaching satiety sooner, that goes favoring, therefore, the control of food intake and the control of body weight. So we're becoming now to that, if you want, personalized nutrition, but I think it's a slightly different concept of what we've been hearing about personalized nutrition. I think that's where nutrition needs to go. Nutrition, yeah, we've seen over the last years that we've been learning about what do we have in common when it comes to nutrition? So how do we digest carbohydrates? How do we digest amino acids? How do we absorb them? And that's fine. So personalized nutrition then says, well, okay, I'm taller, I'm smaller, I'm a runner, I'm a swimmer, and all these differences. Personalized nutrition takes into account this energy expenditure based on the fact that we have the same metabolism. But that might not be true. What actually is happening is that we have a machinery that each one of us has a distinct sensitivity for nutrients, and therefore the capacity to absorb and metabolize those nutrients, and the signal that we're sending to the brain is different. So I think we're going to see nutrition trying to integrate what we have in common, all these digestive processes that we've learned, together with the diversity of the receptors that makes us actually quite different from each other, right? I will spend the last 3 or 4 slides just outlining where we are in extrasensory taste receptors. I've mentioned the important role of taste receptors in the GI tract, but that's by far not the end of the story. Now we know that the taste receptors, taste sensory cells, are present in the respiratory system. A lot of interest has been driven through what they might do in the airways and related to smooth muscle and smooth muscle contraction. I'm going to briefly further elaborate on that a little bit. We know they are present in the central nervous system, in the hypothalamus, for example. Hypothalamus being the main organ in the control of food intake that we know of. Well, taste receptors are there. What are they doing there? Well, hopefully monitoring nutrient levels, glucose, etc., right? They are present in the reproductive system. And actually another one that I'm particularly keen on, and I think has a lot of potential, the immune system. We know that the immune system is actually also expressing receptors, particularly T1Rs and T2Rs that we know of, but also the FFA4, which is actually sensing long-chain polyunsaturated fatty acids. So all this with massive amount of research, and I think we're going to be learning quite a lot on this extrasensory story of taste receptors. That's, by the way, a review that was written by Simon Foster of the School of Biomedical Sciences. And Simon and his main supervisor, Wally, Professor Thomas, they were actually the first group in illustrating the potential relevance of taste receptors in the heart and the cardiovascular system. What they found is a number of also somofactory receptors, but they focused in taste receptors. A number of taste receptors that were present and expressed significantly in myocytes and fibroblasts. And I guess one of the most compelling findings, at least from a nutritionist point of view, is the fact that these receptors in the heart, they were actually responding to the nutritional status of the rats. So that when they were deprived from glucose, you would see an increase in the expression of the receptor, significant increase. Now, last story of my talk today goes to the upper and lower airways. I could have chosen probably other examples, but I just find this to be especially relevant because it switches again what we knew and what we expect from taste receptors, a different story involving the same players, the T2R38 that by now you probably have heard of, hopefully. Now, this is a trachea, so lower airways. This is nasal airways, so upper airways. In both epithelia, you will find quite a very large amount of taste sensory cells, or, well, in this paper they mentioned, they call them solitary chemosensory cells. Same thing. So what are these chemosensory cells doing here? Well, we don't know what they do, but we start having some clues that I will go through. It seems that they do send signalings to the immune system through the trigeminal nerve. And the most compelling story is about how this taste receptor number 38 that we've been talking about, the prop status thing, actually relates to sensing some of the quorum sensing molecules from Pseudomonas aeruginosa, which is a gram-negative bacteria. So these molecules, they're actually sort of indicating population density of bacteria, basically. But what happens is that the receptor number 38 has actually high affinity for those quorum sensing molecules. In fact, they are what? Yeah, lactones, right? So they have a high sensitivity for these lactones. Well, that's if you're a hypersensitive guy. If you're a hyposensitive guy like me, maybe not. And actually, that's what it's shown in the graph. The capacity to clear bacterial infection depends on your genetic background. So if you are a good prop sensor, so you have a very accurate gene number 38, you're capable of kicking the bacteria out through ciliary movement and through the activation of the immune system, as opposed to if you're just a simple AVI like me. So you have to be careful, because we will be more prone to respiratory infection because of the lower capacity to clear these bacteria, and that's the same type of arrangement graphs saying practically the same. The capacity of clear the bacteria is related to PAV, which is hypersensitive to probe, and not to the other two phenotypes. Now, so let me just conclude. I've set the arena of our research group interest in trying to find and document novel paths to explain or to partially or to help explain overconsumption, obesity. We believe that some of the answers of this lack of control of consumption may be laying in our genome, in our polymorphisms, in who we are, in our genes. And that should be, I think, probably the starting of new ways of thinking of nutrition and dieting for human beings, looking at and understanding the differences between us and progressing to what until now has been what do we have in common, which we do have a lot of physiology in common, but then what else is in there? And there are a number of differences that make us quite unique when it comes to the nutritional perspective. And the reason why taste receptors are important is probably because, yeah, they are present in the tongue, and that's why we call them taste receptors. But if somebody would have found them first in the stomach or in the GI tract, would we be calling them taste receptors? Probably not. But since obviously we discovered them in the tongue, we call them taste receptors, but probably a better name would be nutrient sensors. That's what they actually do. They sense nutrients and nutritional status. And the expression of these nutrient sensors, I said, is constant all through the GI tract. But even outside the GI tract, we're talking about important function or potential important functions in the respiratory airways, in the immune system, or in the cardiovascular system. And with that, I would like to thank my team, particularly the data that I've presented today from my team, thanks to Asia and Prits. Also thanks to Nadia, she's been very active in the gene expression work in our team. Thanks to her as well. And thank you for your attention. I'm going to give the floor back to Barbara. If there's any change of question, I'm available. Thank you.